Bone is dynamic. The remodeling process continuously breaks down bone in order to release stored minerals such as calcium. Minerals absorbed in digestion are then used to rebuild bones. 99% of the body's calcium is stored in the skeleton. Remodeling is regulated by two needs. The first is maintaining calcium ion levels in blood, and the second need is that remodeling responds to its bone growth and adaptation to physical stress. Hormones play an important role in the regulation of blood calcium levels and bone growth. Blood calcium is necessary for several physiological processes, including neural processing, glandular secretion, and muscular contraction. Blood calcium is closely regulated between 9 and 11 milligrams per 100 mLs. Elevated levels known as hypercalcemia weaken muscles and produce neurological symptoms. Low levels of blood calcium can lead to muscle cramps and convulsions. The body's cellular needs for calcium may cause blood calcium concentrations to become depleted. To maintain calcium homeostasis in the blood, bone must release calcium ions. Parathyroid hormone, or PTH, secreted by the parathyroid gland increases the number of activity of the osteoclasts. The resulting bone resorption increases levels of calcium ions in the blood. PTH also provokes the kidneys to help increase calcium ions in the blood. First, PTH stimulates tubule cells to limit calcium loss in the urine and step up calcium resorption. Second, PTH stimulates the kidneys to produce the hormone calcitriol, which works in the intestines to increase absorption of calcium from food. That is. Both actions help to increase blood calcium when levels are low or lower than normal. Elevated blood calcium requires the body to store more calcium in bone or excrete more in urine. The hormone calcitonin, secreted by the thyroid gland, promotes the deposition of calcium into bone matrix. The resulting bone formation decreases the level of calcium ions in the blood. Calcium absorption and excretion all help regulate remodeling and growth. Bone growth requires vitamins, minerals, and hormones. Dietary vitamins enter the bloodstream via digestive tract. Vitamin C, along with vitamin K and B12, is needed for the synthesis of collagen. Vitamin A stimulates osteoblasts, the bone-building cells. Vitamin D is needed to form calcitriol, a hormone that increases calcium concentration in the blood. Several hormones influence the activity of bone and cartilage cells. Insulin-like growth factors produced by bone tissue, muscles, and liver promote cell division at the epiphyseal plate and enhance synthesis of proteins needed to build bones. Human growth hormone produced by the pituitary gland stimulates IGF production. Sex hormones produced by gonads, thyroid hormones, and insulin also regulate bone growth. During childhood and adolescence, bone growth occurs in spurts. These spurts are caused by interactions of several hormones, some excitatory, others inhibitory. Insulin-like growth factors, IGFs, are the most influential hormones during childhood. Bone growth production in of IGFs occurring mainly in the liver is stimulated by the human growth hormone. The IGFs travel from the liver to the site of bone growth. Insulin-like growth factors stimulate the formation of cartilage matrix and protein synthesis. During bone growth, high levels of growth hormone increases blood glucose levels, which in turn stimulate the production of somatostatin. High blood glucose stimulates the production of somatostatin in the hypothalamus. Somatostatin inhibits the pituitary gland's secretion of growth hormone. In fact, it is also known as growth hormone inhibiting hormone, or GHIH. Decreased secretion of growth hormone lowers the liver's output of IGFs, slowing bone growth. Bone growth continues when the pituitary gland steps up. Secretion of growth hormone in young adulthood, bone growth typically slows down significantly. Long bones stop growing altogether. Sex hormones, most active during puberty, causes a decrease in the activity of chondroblast, but an increase in proliferation of activity of osteoblasts. As puberty ends, increased ossification at the epiphyseal plate eventually results in the sealing of the epiphysis.